We've arrived at our last parasitic nematode. Trichinosis or trichinellosis is usually asymptomatic if the infection is light. However, gastrointestinal symptoms may be present when larvae invade the intestines. 1 week after infection, larvae migrate to muscle tissue, and more prominent symptoms such as facial swelling around the eyes, conjunctivitis, muscle aches, fever and rashes may be present. Muscles may also increase in size when larvae encyst, and then degenerate, becoming weak later on. There may also be life-threatening manifestations, such as inflammation of the heart muscles, also known as myocarditis, inflammation of the lung tissue, also known as pneumonitis, and central nervous system involvement. An elevated level of eosinophils is also usually present. The transmission route is usually through ingestion of raw, undercooked, or smoked meats that contain encysted larvae. The disease is found worldwide and is most common in Europe and North America. Unlike other nematodes, it has a lower prevalence rate in the tropics. Infected animals are either definitive hosts or potential intermediate hosts, with a second host required to perpetuate the life cycle of this worm. There are 2 cycles of transmission: the domestic cycle, which involves pigs and rodents, and the sylvatic cycle, associated with bears, moose, and wild boars. Humans are accidental hosts. Once ingested, larvae are released from cysts after exposure to gastric acid and pepsin in the stomach. They then invade the mucosa of the small intestines and develop into male and female adults. Female worms release larvae that migrate to muscle tissue, where they encyst and become calcified. They can remain viable for several years. Encysted larvae are spiral-shaped and are quite small, between 1 to 2 millimeters long, depending on whether they are male or female. It is better to use microscopy to view them. Therefore, lab diagnosis involves identification of spiral-shaped larvae in muscle biopsies using microscopy. Alternatively, enzyme immunoassays can be used to detect specific antibodies. Infections can be treated with anti-helminthics. Prompt treatment prevents larvae from becoming encysted in muscle tissue, because once this occurs, it may not be possible to eliminate the infection, and symptoms may persist. Trichinosis can be prevented by cooking pork to at least 60 degrees Celsius and ensuring that meats from other definitive hosts, such as bears and wild boars, are cooked adequately before consumption. There was a large outbreak in 1993 in Dufferin County associated with improperly processed smoked meat from infected wild boars. We will also have a paper next week for your problem-based learning case on trichinosis in bear meat. It was a very interesting outbreak in France that was traced back to a hunting trip in Quebec. These are the reports of nematodes isolated from specimens submitted to the Ontario Public Health Labs. You can see that the most common nematode identified is Ascaris. Pinworms are a close second, then hookworms and Strongyloides. The parasitology kit provided by the Public Health Labs usually has 2 types of containers. One is a sterile empty vial for live cultures, and the other contains a preservative solution of sodium acetate formalin, usually referred to as SAF. Because formaldehyde is toxic, the second vial will have a Wimmis skull and crossbones symbol on the label, and warnings to wash it off if there is accidental skin contact, as well as not to ingest it and to keep it away from young children. The stool specimens are submitted for ONP tests, which are usually microscopy. Sometimes PCR will be carried out for species differentiation.